See carefully. The most cranial part you can see black hairs.
Ahí está como que no ha nacido.
Now here you can see the face.
Así lo ¿verdad?
This is the amniotic sac inside which the baby can move hands as well as legs as well as take out her tongue also.
La mamá ahora.
Sí.
Está tragando líquido.
Está mamando bolsa.
You can appreciate upper limb, lower limb, abdomen, face, eyes, nose, hairs, forehead, lips. Now amniotic sac is removed. See the baby. So you may get a short note on amnion, chorion like that. Okay? So again we will move to the model. This is to clear your idea about the amnion. Amniotic sac enlarges and it occupies the extra embryonic silo. Okay? So extra embryonic silo will not be seen. Amnion will approximate with the chorion. So first there is inside amnion, then there is a chorion. Okay? Why you are not wearing aprons? Okay, now I will allow, this is the first time, but you should wear. Okay? So first there is amnion, then it is approximated with the chorion. So you should know what is amnion, what is chorion. Definitions and whatever along with that you should know about information, and you should write in points. Don't write sentences, and draw whatever diagram you can draw. Okay. So this is the first thing. Now we will go to the models. Now see, this is a triangular disc. In this triangular disc, you can see here blue colored is the amniotic cavity. So this blue colored is the amniotic cavity. So amnion means what? This roof lateral wall along with the extra-embryonic mesoderm. This forms the amnion. Okay? This is a sac. This amniotic cavity will enlarge and embryo is here. So it will encircle all the surrounding part of aspect of the embryo, and it will get connected. That part is known as umbilicus. So this is the amniogenic roof and lateral wall. This is now triangular disc is formed. So this epiblast is known as ectoderm. This is endoderm, and middle pink colored part is the mesoderm. Then this pink colored part is the extra embryonic mesoderm. It is formed by the trophoblast cells. We have seen trophoblast and embryoblast, isn't it? Embryoblast will form this part that is coming from the inner cell mass, and outer cell mass will form trophoblast, from which these extra embryonic mesoderm will be formed. Along with that, it will form syncytial trophoblast, isn't it? So trophoblast, you should remember, it is going to form membranes of the embryo, which will protect, which will nourish the embryo. So this is again yolk sac. Yolk sac is going to form GI tube, isn't it? Now you should know the derivatives of endoderm, ectoderm as well as mesoderm. After this, I will show you PPT. In that I have mentioned all these things. So now if you see inside the amniotic cavity, there is a small groove. This is a primitive groove. Primitive knot is here, which will again show depression. Through this depression, whatever multiplying cells are there, they will enter in between epiblast and hypoblast, and they will form a mesodermal layer. So all three layers are coming from this layer only, epiblast. That you should remember. Now coming to this is a model showing notochordal process. This is a notochord black color. Whatever you are looking, this black color is a notochord. So notochord develops in mesoderm. This one is the amniotic cavity. Above this means dorsal to this embryonic disc is a amniotic cavity, and ventral is yolk sac. Okay, ventral is yolk sac. In between mesodermal bladder. In this one is black colored structure is a notochord which is developing from the primitive knot or primitive node. Here the cells will multiply. They form a globular structure first. Then there will be appearance of pit. And through that pit, the cells will enter in between epiblast and hypoblast. Okay? And then the process will be formed. These cells will move towards the prochordal plate. So from primitive knot to prochordal plate, whatever process, solid process develops, that is a notochordal process. That is called a notochordal process. What is in this white colored structure? That is primitive streak and primitive node. Okay? Primitive streak and primitive knot or node. Both are correct. Coming to the next one, again the development of notochord, very important. You may get a short note on notochord also. Okay, notochord stimulates the development of neural tube, so it is very important. So this is a primitive node where the cells multiply. They enter in between epiblast and hypoblast. First, this structure is a solid structure. So it is called a notochordal process. After that, the canal develops, and it is known as notochordal canal. So notochordal canal has floor, roof, and lateral walls. This is lateral wall, this is floor, and this one is roof. Isn't it? Now floor approximates with this hypoblast. Isn't it? This is an endoderm, which is going to form, still it is not formed, now it is known as hypoblast. So hypoblast and floor, they come very close to each other and perforations are formed in between them. And so amniotic fluid traverses from the amniotic sac through the amniotic canal through these perforations into the yolk sac. Why this circulation is important? Because notochord is developing and all these cells should get nutrition. So the amniotic fluid should enter this space and it should come through the floor also, so that these cells will be nourished. Okay? Afterwards this floor disappears and whole of the amniotic fluid comes here and it enters the yolk sac. So there is a wide opening because floor has disappeared. Whole of this is not seen. Only roof and lateral wall is seen. So wide opening is there. So communication between the amniotic sac and yolk sac becomes too much wide, so that amniotic fluid easily enters the yolk sac. So the diffusion area increases and diffusion becomes more easy. So you should know about this. This is an amniotic cavity. This is a yolk cavity. This is a notochordal canal. Okay, it starts from primitive node to this is prochordal plate. Okay, it is only in between these two. Now it stimulates the growth of neural tube by stimulating the surface ectodermal cells here, which are adjoining to notochord. Okay. So this is an important development of the notochord. Now here also you can see trisomite state. Okay. There is a triangular disc, ectoderm is there, endoderm is there, and mesoderm is there. And in ectoderm you can see a groove. and these cells have become thick, more thick than laterally going cells, isn't it? So there is a thickening of ectoderm adjacent to notochord, because notochord stimulates here these cells. They differentiate, they become more thick, and this is a neuroectoderm. Surface ectoderm differentiates into neuroectoderm. This neuroectoderm, now this groove goes on enlarging, deepening occurs, and these surface cells will get separated. That I have mentioned in the PPT, I will show you. This surface cell will differentiate and they go dorsolateral to this neural tube. Neural tube will be formed. First neural groove and then neural tube will be formed. Okay? And some cells get separated and surface ectoderm again develops as it is. So there will be surface ectoderm, then crest cells, neural crest cells, then there will be a neural tube, and below neural tube will be notochord. Are you getting this? Then laterally, mesoderm again forms the segments. 3 longitudinally running segments are formed by 2 grooves. One groove is here, one groove is here. So paraxial mesoderm is there, then intermediate mesoderm and lateral plate mesoderm. The derivatives of all these you should know. Ectoderm, endoderm, mesoderm. In mesoderm, paraxial mesoderm, which is going to form somite. Then most cranial are somatomeres, that you should know. Then what is formed from the intermediate mesoderm and what is formed from the lateral plate. That is derivatives of all these you should know. Then this is a somite stage. But whatever this slipper-like structure is there, this slipper-like structure is nothing but the neuroectoderm. Neuroectoderm first forms a neural plate because only surface ectodermal cells become thick and they form a plate. Now this plate formation is pre-somite stage. Here what has happened? Groove is formed as well as folds are there, lateral folds are there. These are folds. See, in the middle there is a groove and these two are folds. So this is a somite stage. Somite stage is from 20th day to 30th day. Now neural plate formation is pre-somite, means it is from 16th day to the 19th day. Okay, 16th to 19th is a pre-somite stage where the neural plate is formed. The neural groove and neural folds are formed in the somite stage on the 20th day, 21st day and 22nd day. Okay, 20, 21st and 22nd. Neural groove and neural fold is formed. Now, from 20th day onward, we count as a somite stage. 20th to 30th day are the somite stage. So, after this neural fold formation, now these folds unite in the midline and they form a neural tube. Neural tube is formed on the 25th day. Okay? It is these two edges unite in the middle part first. Then this union extends cranially as well as caudally. In this way, neural canal is formed. It is open anteriorly and posterior. Cranially and caudally it is open, and that is anterior and posterior neuropore. Now this union goes on cranially, and anterior neuropore is closed on the 25th day, and posterior neuropore is closed on the 28th day. That you should remember. Okay? So what is this? These black colored small cavities are shown. These are the cavities in the mesoderm. These are cavities in the mesoderm, extra-embryonic coelom. These cavities unite and they form a single large cavity. Okay? Intra-embryonic coelom. It is an intra-embryonic coelom which forms a horseshoe-shaped cavity. Okay? This horseshoe-shaped cavity at the cranial end in the midline forms the cardiogenic area that we will see afterwards. Remember that this is in the mesoderm, this cavity. But blue colored structure is the amniotic cavity. This is a cut edge of the amnion. It will be like this. Okay? And this will be the yolk sac. This is the connecting stalk. You should know about the connecting stalk also, which is going to develop in the umbilical cord. What are the contents of umbilical cord? You should know. Yes, Wharton's jelly. This connecting stalk will be surrounded by this amniotic cavity as it enlarges, isn't it? So amnion will be the outermost layer of the umbilical cord. And inside what you get? Extra-embryonic mesoderm, which undergoes mucoid degeneration and it forms a Wharton's jelly. Wharton's jelly. It will support the structures which are present in the umbilical cord. Which are the structures in the umbilical cord? Yes, there are two umbilical arteries, one umbilical veins. Along with that you get, yes, allantoid diverticulum, part of the yolk sac will be there. Then from midgut loop will be coming, which is known as physiological hernia, that you will learn in the development of alimentary canal. So these are the structures present in the umbilical cord. Two arteries, one vein, allantoid diverticulum, extraembryonic mesoderm, which undergoes mucoid generation and forms mucoid degeneration and forms Wharton's jelly. Okay, along with that you should mention about vitelo-intestinal duct or yolk stalk. Isn't it? Yolk stalk get pinched and small part which is at the periphery may degenerate and remaining part forms the intestine, foregut, midgut and hindgut. Isn't it? And these two parts are joined by the yolk stalk, or it is known as omphaloenteric duct. Okay? So, we will see that part. I will show you. Again, this is a trilaminar germ disc. You can see here amniotic cavity, yolk sac, and in between will be the trilaminar germ disc. Here is a primitive streak, primitive node. This area is known as procaudal plate. Procaudal plate. So, caudal to this here will be the cloacal membrane. Cloacal membrane and procaudal plate are the two areas where you don't get mesoderm. This is very important. Okay. Now see here. This is a yolk stalk or omphaloenteric duct. Okay. Now this yolk sac, because of the tail fold and head fold, it is getting pinched here. So this part gets separated from this sac. Okay, this sac reduces in size and disappears usually. Okay, and this part forms the foregut, midgut and hindgut. The part which is enclosed in the head fold, that forms a foregut, so this part. Then part which is in the tail fold, this part. This will form a hindgut, and remaining part which is having wide opening into the yolk sac, that will form a midgut. What are the derivatives of foregut, midgut, hindgut? We will see in the picture. Now this head fold, because of this head fold, what happens? This is a procaudal plate or buccopharyngeal membrane. It is called as buccopharyngeal. There is a depression formed here which forms the oral cavity, and this membrane is cut at one stage by which the embryo swallows the amniotic fluid. Embryo swallows amniotic fluid. It passes through kidney and comes out as a urine in the again same amniotic fluid. Urine also enters, but this urine doesn't contain any excretory material. It is only water. So it swallows it. It is excreted as urine, but it is containing only water. So foregut, midgut, hindgut. This blue part is a neural tube. It is a neural tube. This part will form a face region. Okay. Next will form a forebrain, midbrain, hindbrain, and then this narrow part will form a spinal canal. It will form a spinal canal. Now this is a tube, so canal is already there. Whatever canal is in the brain, it will form ventricles. Not only this, the small canal at the last end will form a cavity of terminal ventricle. Terminal ventricle is in the phylum terminal. Do you know phylum terminal? Phylum terminal is the last slender part of the spinal canal. In that phylum terminal, there is a proximal bulge. Slight bulging is there, which contains a cavity, that is terminal ventricle. So this cavity is represented here by the canal at the distal end of the neural tube. Okay? This canal will form a canal of the spinal cord, and remaining will form a ventricles of the brain. Forebrain will be having lateral ventricles. In this way, each ventricle, lateral ventricles are there, fore ventricle is there, both ventricle is there. So the aqueduct is there, aqueduct of Sylvius is there. So these are all canals which are coming from this canal. That is canal in the neural tube. Now anterior this neural tube is closed, which in the adult is represented by the linea terminalis. It is called as linea terminalis, which is the anterior-most wall of the third ventricle. Anterior-most wall of the third ventricle. And here it forms the terminal ventricle. So most distal part of the terminal ventricle will be formed by the posterior neuropore, which is closed at the 20th day. Anterior neuropore is closed at the 25th day. Next to that, this one is a, what is this? Notochord. It disappears and forms nucleus pulposus and apical ligament of Dens. Okay? Some cells may remain in the nasopharynx and they will form a malignant tumor that is known as chordoma, which is more common in the adult male. Okay? So this is the applied aspect. Then you get this yolk sac, which can be divided into foregut, midgut, hindgut, which we will see the derivatives afterwards. Now this is a allantoid diverticulum. First it was coming in this direction, but because of the movement of this, like this tail fold, this allantoid diverticulum is coming on the ventral aspect of this foregut. Okay, foregut not in hindgut. On the ventral aspect of the hindgut because of the movement like this. Okay. Now this allantoid diverticulum has two parts, proximal part and distal part. This proximal part is absorbed in the cloaca. Here. Okay. Which will form a urinary bladder. Okay. Proximal part. This part is absorbed in the urinary bladder. And distal part becomes fibrous. And it gets connected to the umbilicus. Umbilicus is developed here. This whole region will develop into umbilicus. It will get narrowed because of the deepening of these folds. But umbilicus is here and this distal end is connected to umbilicus and proximal end is connected to the apex of the bladder, urinary bladder. Okay? So this fibrous cord is called as uracus. It is called as Uracus, which connects the apex of the urinary bladder to the umbilicus. It is a fibrous cord. Okay, so this is. Now this is included in the umbilical cord, allantoid diverticulum. Then this is a omphaloenteric duct. Okay, enteric means intestine, omphalic means this sac. So, yolk sac. This will reduce in size. And this is known as tertiary yolk sac or definitive yolk sac. In many books, it is mentioned as definitive yolk sac. Okay. Now, because of the head fold, what happens? Buccopharyngeal membrane is here, which was actually relations now have changed. See, this is coming cranial to the pericardial cavity and cardiogenic area, which was caudal first, isn't it? Caudal in position, now it is coming cranial. So, this is a pericardial cavity which is on the dorsal aspect of the, this is cardiogenic area which is on the dorsal aspect of the pericardial cavity. This one is pericardial cavity, this is cardiogenic area which is shown by the red color. When the embryonic disc was flat, no folding was there, that time this was ventral to the pericardial cavity. Isn't it? It developed in the splanchnopluric mesoderm. As the head fold occurred, the position changed from ventral to dorsal. Okay. Now distal to this will be here septum transversum. Septum transversum is the most cranial structure in the embryonic disc. Now it has come distal to this pericardial cavity and cardiogenic area. Is it clear? These are the changes taking place because of the head fold. So septum transversum is here. What develops from septum transversum? Central tendon of the diaphragm and liver. Okay, part of the liver actually. So this is about this folding of the embryo. Now this is the one showing primordial cardiovascular system. We are going to start with cardiovascular system. Previously I have completed one lecture on this, but I don't know how much I have seen that lecture because of the less capacity of entering into that procedure. So coming here, this is called embryo developing. This is the umbilical cord. This one is umbilical cord. So you get here umbilical vein, umbilical artery, black color whatever is shown. Okay, coming to these veins first. Blue colored are veins, isn't it? Now this is a developing heart. Heart is developing, heart tube is there. So this has developed from the ventral aorta. Ventral aorta are 2 in number, which fused and form a single tubular structure which has 4 parts: bulbous cordis, primitive ventricle, primitive atria, and sinus venosus. So 4 parts will be formed from this. Sinus venosus is this because it drains these veins. Okay? 3 veins are important here. One is the vitelline vein coming from the yolk sac, vitelline vein. Then umbilical vein is there. This one is umbilical vein. It is shown red in color because it carries oxygen and nutrition from placenta to baby. So this is a umbilical vein. See, always students confuse between their relations. Most medial is vitelline. It is not. One model is there. Okay, we will see it afterwards. Most medial is vitelline. Then lateral to that you will get umbilical vein, and lateral to that is a common cardinal vein. Common cardinal vein. Again I will repeat: vitelline vein, umbilical vein, and common cardinal vein. Only this part is common cardinal because it gets blood from the posterior cardinal vein and anterior cardinal vein. Okay? Posterior cardinal and anterior cardinal will unite here and form a common cardinal vein. Okay, so this is the venous end of the heart tube, and here is the arterial end. This one is the dorsal aorta. This is a dorsal aorta. It gives here small branches which unite with the aortic arch here and aorta here, and they will form aortic arches. Can you see these small vessels? These are aortic arches. These are called as aortic arches. Here laterally it gives one branch which goes to the yolk sac, and this is a vitelline artery. Again posteriorly it gives one branch or continues here into the placenta as umbilical artery. Okay, so these are important: aortic arches, vitelline artery, and umbilical artery. There are 2 umbilical arteries, but only one umbilical vein. Right umbilical vein disappears. Only left is left. You can remember like this: left umbilical vein is left. So here you get umbilical artery, umbilical vein, vitelline artery, vitelline vein. Here is aortic arches. This is a dorsal aorta. You should know this. This is the anterior cardinal vein, posterior cardinal vein, common cardinal vein. Okay. This is the umbilical vein. This one is vitelline. should be able to identify each artery and vein. 3 veins, 3 arteries are there. Okay, we will see derivatives afterwards, but important is this. Okay. So this whole is yolk sac. Okay. This is also an important model because here they have shown neural crest cells. This is a neural tube formed from the surface ectoderm. So neural cells are neural tube is ectodermal in origin, remember. Now, during the separation of this neural canal, formation of neural tube, some cells of the fold, apex of the fold, they get separated and they lie on the dorsolateral side. Actually, they are not continuous like this. One bunch is here, one bunch is here. Dorsolateral to this neural tube. These are the neural crest cells. This is surface ectoderm. It again unites and forms continuously one layer here after separation of this neural tube. Then you get here, this is notochord, ventral to neural tube. There is a notochord. Okay? These are the paraaxial mesoderm. Okay? Now this is the yolk sac. These two will be the lateral folds because the cross section is this. This is a cross section of the embryo. So these two are the lateral folds. So here, if intraembryonic coelom is there, this will be the extraembryonic coelom, which are united with each other through this orifice. Okay? As the lateral fold deepens, the intraembryonic gets separated from extraembryonic coelom. And this intraembryonic coelom will form pleural cavity, pericardial cavity, and peritoneal cavity. Okay? Extraembryonic coelom will disappear because that space will be occupied by amniotic cavity. Okay? Now this is the lateral view of the embryo, 28 days. Okay? So they are about 1 month. So this is the head region. These are the somites, you can see nicely. Then here is the upper limb. Here is the lower limb. This is the umbilical cord. Okay? Now what is this? These are the pharyngeal arches. Pharyngeal arches. Whatever arteries enter, they are called as aortic arches. These are pharyngeal arches. Now what are these bulges? Here the heart develops. This is the bulge of left atrium and this is the bulge of left ventricle. Is it clear? Again I will say, this is the head region. Here will be the development of face. Okay? These are the pharyngeal arches. Somites. Okay? Upper limb bud, lower limb bud. This is the caudal end, umbilical cord, left atrium bulge, left ventricular bulge. Okay, now we will see PPT in which some important comments are there. Models,
मैडम कितना है? कितना है?
So you should know derivatives of ectoderm, endoderm, and mesoderm. Here in short, I will enumerate these factors. This is not a part of understanding. Only you have to by heart these names. Okay. Skin and appendages. What are the structures coming from ectoderm? In skin you get this anyone knows? You get epidermis, nails, hairs, sebaceous glands, sweat glands, erector pylori muscle. Okay, these are the structures of the skin derived from the ectoderm. If we come to eye, various structures from ectoderm are lens is there, anterior epithelium of the cornea is there. Okay, then ciliary muscles are there, conjunctiva is there. Along with that, you get lacrimal gland and nasolacrimal duct, which are ectodermal in origin. In ear, you get utricle, then semicircular canals, outer part of the tympanic membrane, outer epithelium of the tympanic membrane, along with the lining of the external auditory meatus. These are ectodermal in origin. These are ectodermal in origin. Then coming to the nose. In nose you get lining of the nasal cavity. Nasal cavity is connected to paranasal sinuses. So lining of the paranasal sinuses. Then olfactory bulb and olfactory nerve. These are ectodermal in origin. Olfactory nerve is the first cranial nerve. Isn't it? It is a first cranial nerve. So these are coming from the ectoderm. Then oral cavity and GIT. In oral cavity, extreme ends towards the mouth as well as end of the anal region are coming from ectoderm. Towards the mouth, which area? Epithelial lining of the anterior 2/3 of the tongue. Then epithelial lining of palate as well as lateral wall of the oral cavity. Okay. Then parotid duct and parotid gland. These are coming from the ectoderm. Okay. Then extreme last part means end part of the anal canal is ectodermal in origin. Then urogenital system. In urogenital system again you get the external vaginal structures. For example, labia minora, labia majora. These are covered by the epithelium which is ectodermal in origin. And last part of the penile urethra is ectodermal in origin. Okay, so these are the ectodermal structures. You should be able to enumerate these. Yeah, both like so next is endodermal derivatives. Endodermal derivatives, which is lining the yolk sac. So embryonic disc is there. So hypoblast forms the endodermal layer. That endodermal layer will give us some structures of the oral cavity and GIT. In oral cavity, we have mentioned that anterior-most part is ectodermal. Remaining all part is endodermal. For example, lining of the posterior 1/3 of the tongue, then floor of the oral cavity. Okay, palatoglossal fold and palatopharyngeal fold, which are surrounding the palatine tonsil. Anteriorly there is a palatoglossal fold, and posteriorly there is a palatopharyngeal fold. In between these two folds, there are palatine tonsils. What general people mention as tonsil, that is palatine tonsil, because there are other tonsils also. So these are the palatine tonsils in between palatoglossal and palatopharyngeal folds. These folds are like this in the nasopharyngeal posterior part of the oral cavity. Okay. In between this, there is a palatine gland or palatine tonsil. So these folds are lined by the epithelium, which is endodermal in origin. Then endodermal lining of whole of the GI except last part of the anal canal, which is ectodermal. Okay. Now some derivatives, because some buds are coming from the GIT which are endodermal. For example, pancreatic bud is there. So in pancreas you get islets of Langerhans. Then there is a exocrine part also for the pancreas. These are the acinar cells as well as duct system of the pancreas is endodermal. Okay. Along with this, liver bud comes from this junction of the foregut and midgut, and this bud develops into the hepatic cells as well as duct system of the liver. So these are all derived from the endoderm. Next is respiratory system. Now foregut gives one bud which develops into the trachea, bronchial tree, etc. So lining of all these trachea, bronchial tree, alveoli, all are endodermal in all are derived from the endoderm. Then ear. In ear, middle ear cavity is lined by the epithelium which is developed from endoderm. Along with that eustachian tube which connects middle ear with nasopharynx, that is also having a lining coming from endoderm. Okay, middle ear cavity and eustachian tube. Then endocrine glands, thyroid, thymus, parathyroid gland. These are the endocrine glands, and some parts of them are endodermal. Which parts? If you take a thymus, there are essence corpuscles. You will see in histology how it looks. Essence corpuscles along with the reticuloepithelial cells of the thymus are endodermal. Okay. Then if you come to the thyroid, you get thyroid follicles. And these follicles are lined by the follicular cells which are endodermal. Then in parathyroid gland, there are principal cells and obsequial cells which are coming from the endoderm. Then urogenital system. In urogenital system, you get a lining of the urinary bladder which is endodermal. Lining of the vagina which is endodermal. Now in lining of the urinary bladder, it is not lining trigone of the bladder, but remaining part is endodermal. Okay, trigone is mesodermal in origin. So, lining of the urinary bladder, lining of the vagina in female, lining of the urethra, whole urethra in female, and lining of the most of the penile urethra in male. Because penile urethra at the distal end is having a ectodermal origin. And remaining part is endodermal. Okay. So after this you get derivatives of mesoderm, paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. In paraxial mesoderm, you get skeletal muscles of the trunk, skeletal muscles of the limb, skeletal muscles of the head. These are coming from the paraxial mesoderm. Along with that, extraocular muscles of the tongue are coming from the paraxial mesoderm. Is somite, what you described. You may get a short note on somite. It's a very important topic. So these are the derivatives of the paraxial mesoderm. We have covered bones of the axial skeleton. For example, vertebrae, ribs are coming from the paraxial. Along with that, cranial bones, are also coming from the pharyngeal mesoderm. Along with that, there is a dermatome which gives dermis as well as dura mater. There are three coverings for the spinal cord and brain. Isn't it? Pia mater, arachnoid mater, and dura mater. Dura mater is mesodermal in origin, but pia mater and arachnoid mater, which collectively called as leptomeninges, these are coming from neural crest cells. Remember, these are coming from neural crest cells. Neural crest cells we have seen already, isn't it? They are present on the dorsolateral aspect of the neural tube. Now coming to the intermediate mesoderm. Intermediate mesoderm gives origin to the kidney, ureter, then trigone of the bladder. We have already mentioned that urinary bladder is lined by the endoderm. Lining is coming from endoderm, except trigone. This trigone is coming from the intermediate mesoderm. Okay. Along with this, you get gonads, testes, ovaries, and their ducts. Testes, ovaries, and their ducts. Ovaries are having ducts which are fallopian tubes, and testes are having ducts to carry spermatozoa. And these are epididymis, vas deferens, and one duct is there. Common ejaculatory duct is there. That is coming from this intermediate mesoderm. Then coming to the lateral plate mesoderm. Lateral plate mesoderm develops a cavity inside, isn't it? It is having somatopleuric mesoderm and splachnopleuric mesoderm. Towards the yolk sac is splachnopleuric mesoderm, and towards the amniotic cavity there is a somatopleuric mesoderm. Now, what develops from this? From lateral plate mesoderm, you get bones of the upper limb along with the sternum. We are not mentioned in axial skeleton, which is coming from paraxial mesoderm, about sternum. Paraxial mesoderm forms vertebrae and ribs, but this lateral plate mesoderm forms sternum as well as bones of the upper limb. Okay. Then there is a cavity which forms pericardial cavity, peritoneal cavity, and pleural cavity. Intraembryonic coelom. We mention it as a intraembryonic coelom, which forms three important cavities in our body. Then this lateral plate mesoderm forms a covering of intestine, except epithelium, which forms all layers. For example, lamina propria. Then muscularis mucosa, submucosa. Then muscularis externa as well as adventitia. All these layers are coming from lateral plate mesoderm. Then it forms two important systems, cardiovascular system and lymphatic system. We have seen that cardiogenic area develops in the splachnopleuric mesoderm, isn't it? In the midline, at the most anterior end of the intraembryonic coelom, isn't it? This is splachnopleuric mesoderm, which forms a heart, which forms a spleen. Okay. So, two important systems, cardiovascular system and lymphatic system, coming from the lateral plate mesoderm. Some important organs such as suprarenal gland, spleen, as well as larynx. In larynx, you get some cartilages. These cartilages of larynx are also coming from the lateral plate mesoderm. Some cells are also there, which are coming from this. These are microglial cells, Kupffer cells, which are coming from the lateral plate mesoderm. So this is very important. You should know derivatives of each: ectoderm, endoderm. Yes, Suraj?
The mostly correct.
Huh?
Most like that's all right. Side side correct. Okay, so this.
This also we have seen implantation of So we are seeing this implantation, primary, secondary, and tertiary yolk sac, along with this tertiary yolk sac is also known as definitive yolk sac. Yes, this model we have seen just now. Amniotic cavity is there, extra embryonic mesoderm we can see, amnion is there, then primitive pit, primitive node is there, primitive groove is also seen, and this is a cross section. See how they have shown primitive pit and primitive node. It is not cranial or caudalate, but it is a cross section. Then trilamellar germ disc is seen, umbilical vesicle. Next. Okay, so this is embryonic endoderm, mesoderm, and ectoderm also you can see. This also we have just seen. Floor of the amniotic cavity, white color structure is the procaudal plate or precaudal plate. Then primitive pit is there, primitive groove is there, which is shown by the white color structures. Black color is notocaudal process and cut edge of the amnion and connecting stop. Okay. So this model also we have seen how umbilical cord is formed. The last figure you can see that large one. In that you can see adenoid diverticulum, small bulge, and large one is the vitalointestinal duct or omphaloindric duct, and small part which is pinched off from the gut tube is definitive yolk sac. So here also you can see umbilical cord and its contents: primary mesoderm or extramarital mesoderm, vitalointestinal duct, adenoid diverticulum, umbilical vessels, and herniation of mid-gut tube, which takes place in the 10th week. Afterwards, it is taken inside, so it is not seen afterwards. So formation of primitive streak is shown. You know this, so I will not mention in detail. So here is the gastrulation process. How primitive streak multiply, cells in the primitive streak, primitive node multiply and spread all over in between hypoblast and epiblast, that is shown. See the position of the cloacal membrane, which is most caudal structure. So this is a trilaminar disc. In this you should know about the paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. Okay, that green colored is lateral plate mesoderm. Inside that violet colored is intermediate mesoderm. You should be able to identify procaudal plate, cloacal membrane. Okay. Then notocaudal process, which is shown as a dotted line, and yellow colored midline structure is neural tube. Okay. So these we have seen many times. We will not go into the details. This also the same model. You can see this is a trilaminar disc. You should be able to identify neural groove, neural fold, ectoderm, mesoderm, and endoderm. Okay. Then paraxial mesoderm. In the middle is notochord. That also you should be able to identify. Intermediate mesoderm and lateral plate mesoderm will also see. Here is the formation of the neural tube. See in the middle picture how it is. The folds are joining, and it takes place in the middle part. Then it extends cranially and caudally. Anterior neuropore and posterior neuropore is also shown here very nicely. So this is the formation of neural tube, which is known as neurulation. Don't confuse between the neuroenteric canal and neuration. These are two different structures developing. Okay. So these are the structures: cardiogenic area, neural plate, neural groove, neural fold. Just we have seen this below its places cuttage of amnion and connecting stalk, which develops into the umbilicus. All information is also we have taken this many times. So this is also important. Modern blue color is the neural tube. Then there is a notochord deep to that. Then there is a primitive gut which is going to from foregut, midgut, hindgut, and then omphalomesenteric duct or omphaloenteric duct. Then you get a yolk sac, head fold, tail fold, and the structures enclosed in it. So this is the buccopharyngeal membrane, foregut, midgut, and hindgut. From hindgut, allantoid diverticulum is coming. We will see afterwards all the derivatives of this. Okay, derivatives of this we will see next time. This is a theoretical part. We have covered this also. Somites are there, pharyngeal arches are there, bulging of the left atrium, bulging of the left ventricle, upper limb bud, lower limb bud, umbilical cord that you should be able to identify. So we will cover this afterwards, but whatever is covered, try to remember the structures.
Lupa,